Hey Setterton, this is Mr. Duvik, and today's YouTube lesson we're going to be talking about our last topic for the year, protein synthesis. Protein synthesis is essentially how proteins are made, okay? And those proteins are super important because they code for our traits that we express. Now what you need to know about protein synthesis is that there are two main stages that we'll talk about later. There is transcription and translation. Transcription is literally where we go from DNA, okay, and we go from DNA to RNA, and that RNA molecule, which is a copy of DNA, is then going to be translated into proteins, okay? So again, if you look at the diagram here, we are looking at a specific sequence in DNA. This sequence is going to be copied into RNA, and that RNA is then going to go to a ribosome to be translated into proteins. And these proteins, okay, are essentially going to express our traits. So that is protein synthesis. So in protein synthesis, we are going to find out about a new molecule. Okay, and that new molecule is called RNA, or ribonucleic acid. Sounds very similar to DNA, but it is slightly different. So what is RNA? RNA is a molecule that is responsible for transferring genetic information from the DNA to the site of protein synthesis, okay, and that is essentially a ribosome. Okay, so essentially what RNA is, it's a copy of DNA, okay, that is made inside the nucleus, and then once made is going to leave the nucleus and it's going to go to a ribosome to be translated and made into a protein. Okay, so here I have just a simple kind of a comparison chart of DNA versus RNA. DNA is called deoxyribonucleic acid. It's a deoxyribose sugar. Okay, it's double-stranded. The base pairs in DNA are GCAT, guanine, cytosine, adenine, thymine, and we call DNA the master plan. Okay, because if DNA gets damaged, then that DNA molecule is forever damaged. There's no way of changing it or fixing it. However, on the other side here, we have RNA, which is ribonucleic acid. Okay, the sugar used in RNA is just a simple ribose sugar. The structure is single-stranded. Okay, so because I've mentioned before, RNA is a copy of DNA, RNA is only going to be a single strand, and that single strand is going to be a copy from a specific sequence from our DNA molecule. The nitrogen bases are going to be GCal, so this is a little change up here. So the nitrogen bases in RNA are going to be guanine, cytosine, adenine, and uracil. So no longer will we have thymine in RNA. So that's a big change up there. Okay, and then the analogy for RNA would be like the blueprint, because RNA is a copy of DNA. It's okay if we damage this, or it's okay if something happens to RNA, because we can just keep making copies of that specific sequence using our DNA. Okay, so RNA is going to be like our copy. It's going to be like our blueprint. So here you have your DNA molecule here, double strand. Then you have your RNA, which is simple single-stranded DNA, or RNA molecule, excuse me, and it's a copy of a specific sequence from your DNA molecule there. So in terms of RNA, there's three different types that we need to go over in order to fully understand the actual process of transcription and translation, which are the two stages in protein synthesis. Okay, so the first type of RNA that we're going to talk about is called the messenger RNA. And the messenger RNA carries a specific copy of a genetic sequence from the DNA in the nucleus to the ribosome in the cytoplasm. So essentially what the mRNA is it's a copy of a very important DNA sequence called a gene, okay, that codes for a very specific protein. So copy of a very important sequence, it's a message, okay, and this very important message from that DNA sequence is going to be made into a very specific protein, and that protein is going to carry out a trait. So that's what a messenger RNA is. It's just a copy from a DNA sequence that codes for a protein. This mRNA is then going to go, once it leaves the nucleus, it's going to go and find our rRNA. Okay, and essentially what rRNA is, it's our ribosome. okay? And what you need to understand is that all ribosomes are made up of multiple subunits, okay? And those subunits are called rRNA. So rRNA makes up a ribosome with other proteins, okay? And our rRNA, I know that's a tongue twister, is going to be the site of protein synthesis, okay? So here's our ribosome. You can see the large subunit, the smaller subunit. And essentially, when we want this mRNA sequence, okay, to be translated, where this mRNA is going to go is it's going to go right in between these two subunits, and it's going to be translated, okay? Meaning it's going to go from this message into amino acids, and then those amino acids are going to bond together to make a functional protein, okay? So essentially, this is going to be the site of protein synthesis. It's where messenger RNA is going to go to be translated. Last but not least, our third type of RNA is called a transfer RNA, which is called, or we kind of just say tRNA for short. And tRNA carries amino acids to the mRNA in the ribosome to form a polypeptide. And now I keep saying polypeptide. For those who don't know what it is just many amino acids bonded together, and essentially many amino acids bonded together is a protein. So really a polypeptide is a protein. Okay, but essentially what our tRNA does is it carries these, we call them anticodons, okay? And these anticodons are going to be complementary to the base pairs on the messenger RNA. And essentially what this tRNA is going to do is it's going to carry a very important amino acid, all right, to the ribosome, and it's going to drop it off using these base pairs, okay? So essentially what you need to know for transfer RNA is that it carries amino acids to the mRNA in the ribosome to begin to form a functional protein, okay? And we'll get into more of that, okay? But literally all transfer RNA does is it goes into the cytoplasm of the cell, it finds the correct amino acid that is needed in the mRNA to make a protein, it picks it up, okay, and then it's going to go to the ribosome. It's going to use its anticodon to bond to the codons on the messenger RNA, and then once it bonds, it's going to drop off that amino acid. This tRNA structure right here that kind of looks like a tRNA is going to leave and go back into the cytoplasm to continue picking up amino acids to then bring down to this ribosome to make a protein. So now what we're going to do is we're going to compare protein synthesis to building a house. So here's a simple analogy that I really think is helpful in understanding protein synthesis. Okay, so say I want to build my dream house here. Okay, I need to go through several steps in order to begin building it and get my dream house, okay, that is fully furnished and everything. So what I need to do in order to make this dream house is I need to first come up with a master plan, okay? So I'm going to choose a contractor, I'm going to sit down with that contractor, and we're going to devise together a master plan. And that master plan is going to have essentially what my house looks like, the workers needed, the materials needed. It's going to need the square footage, the dimensions, where all the rooms are going to go, what the kitchen's going to look like, and so forth, okay? So essentially, the master plan is going to be the original copy of the layout of my house and what it's going to look like, okay? And it cannot get damaged, because if me or a construction worker or the contractor loses this master plan or damages it, then that's going to change the whole setup of my house, and that's not good, okay? Also notice that the master plan has a gate around it, okay? And that's important so nothing damages that master plan. Next, okay, I'm going to talk about construction workers, and the construction workers are going to act as the ribosome, okay? And essentially what the construction workers are going to do in this process is they're going to take the instructions to make this house, and they're going to begin to build it, okay? So essentially, they're going to be the site in which my house is built. Next, okay, each construction worker is going to need a copy of my master plan, because remember, my master plan cannot get damaged. So we need to make copies of that master plan. So what do we call copies of the master plan or the original copy? We call them blueprints, and blueprints are just copies of the master plan, and it's okay if we damage them, okay, or we lose them, because we can just make more copies over and over and over. Okay, so essentially why we need these blueprints is because each construction worker needs a blueprint so they know exactly what they're doing. They know exactly the dimensions of the project they're working on and the materials they need. Okay, so that's the blueprint. Next, we need materials. Okay, so the materials are going to be needed in the process for building this house. Construction workers are going to need to know the materials in order to build whatever they're building. Okay, and essentially when the construction worker begins to put together all the materials, what he's going to then get is a finished product, which is my dream house. Okay, so let's now look at it kind of in the biology viewpoint. So here are master plan, or original copy of my house, is going to be considered to be the DNA. And the reason is because the DNA contains all the important instructions to make a protein. Okay, all these base pairs code for different proteins, and it's super important. If this DNA molecule gets damaged, it gets lost, that's not good, and that's how mutations arise. Okay, also the DNA is protected in the nucleus, just like my dream house was protected around that gate. Next, we have our RNA. Our RNA is just copies of a specific DNA sequence. Okay, and essentially the important part that you need to understand is that RNA, okay, can leave the nucleus because it's just a simple copy. We can make more and more copies over and over and over again if we lose this mRNA sequence. Okay, so essentially this RNA sequence, okay, is going to be given out to all the construction workers so that they know specifically what they need to do in order to build my dream house. Next, we have our ribosome. So our ribosome acts as our construction worker. Our construction worker, okay, is literally going to take the blueprint, the materials, and what it's going to do is it's going to begin to build, just like the ribosome does. It intakes the mRNA sequence, and it's going to essentially translate this message into a protein. Last but not, or almost last but not least, we have our amino acids, and our amino acids are going to be like our materials, because as we begin to take more and more materials and build them together and combine them together, we're going to make my finished product, which is my house, and in this case it's a protein. So the more and more we build and we bond together amino acids, the more we get closer to making a fully functioning protein. And once we have all the amino acids bonded together that are needed to make a protein, what we're going to form is a fully functioning protein. Okay. All right, so what we have here is called the central dogma theory, and so this is essentially a theory created by Francis Crick, who was also one of the scientists who discovered the structure of DNA. Okay, and essentially Francis Crick wanted to know how the genetic code in DNA made a protein, because that is pretty, significant and crazy to understand. And so what essentially Crick did was he said that DNA has the instructions for the order of amino acids, which determines the protein that make up traits of any organism. Okay, and again, traits are hair color, eye color, things like that. Okay, so essentially what the central dogma states is that it's stating that when we are trying to create a protein, where we're going to start first is at a specific gene. Okay, and remember, a gene is a location on a DNA molecule that codes for a protein. Okay, so what we're going to do is we're going to look at this DNA molecule and we're going to look at this gene right here, because this gene codes for this protein. And in order to get to that protein, what we need to do is we need to make a copy. Okay, so we're going to make a copy of this DNA sequence right here, okay, to form mRNA. So the actual process of going from making a copy of DNA into mRNA is called transcription. Okay, then what we're going to do with this mRNA is we are going to send it through and to a ribosome to be translated into a protein. So essentially DNA to mRNA is called transcription, where we're literally making a copy. Okay, and then from mRNA to a protein is called translation, and this mRNA is going to go to a ribosome and it's going to be translated into a fully functioning protein, and that's the central dogma theory.